I am very pleased to present to you our second series of MesoTV. MesoTV is a program that was created by the Mesothelioma Applied Research Foundation in response to the need to cancel our International Symposium on Malignant Mesothelioma. We recognize that many were seeking information about treatment, the science behind malignant mesothelioma, news of the community, and information about the workings of the Mesothelioma Applied Research Foundation. This program has been made possible by our generous supporters and sponsors. Thank you so much. It is so important for us to be able to bring all this pertinent information to the community that we serve.
Good afternoon, Dr. Zauderer. Thank you so much for joining us today in this latest episode of MesoTV.
Thanks, Mary.
Thank you. So I'm not sure if you're aware, but there's so much buzz right now going on in the community about these CAR T-cell trials. And I know that, you have a few that are, underway at Memorial Sloan Kettering. And I wondered if we could focus today on the Dietary trial, as I know you're very intimately involved with that trial. So could you tell us a little bit about CAR T-cell to begin with?
Sure. It's an involved process, but basically it's what we call a cell therapy. It's sort of a living treatment that's given to you. And so the way a CAR T-cell is made is a person gets hooked up to a machine, almost like they're giving a blood donation. But instead of just the blood going into a bag and being taken away, it's connected to a machine at the bedside that pulls off those immune cells that we want to use, those T cells, and it gives you back the rest of the blood. So you shouldn't feel lightheaded, need the juice and the cookies, because you're getting all of your blood cells back. You're just not getting some of these immune cells. We then take those immune cells to the lab where we treat it with a viral signaling vector that basically inserts some new coding into the cells. And basically the way I think about it is, instead of those cells that might have been intrinsically set up to target, a different antigen, something to react to something else, what this treatment does is it inserts a sequence that tells that cell, oh, you should focus on mesothelin. Mesothelin being a protein on the surface of mesothelioma cancers commonly. And so what happens is it's almost like a boot camp, and it takes these sort of cells and it teaches them mesothelin shouldn't be here. This is what you should attack. This is what you should go to. And then that takes a period of time to transform those cells, and then we grow them out. We replicate those cells, and then we freeze them. And then when a person is ready to get this treatment back, in our particular trial, the cells are given back into the chest cavity. So you either need to have a pleural effusion, a pleurix catheter, or an area of disease that we can access and inject. And we work really closely with our interventional pulmonology team and the interventional radiology team to look at the pictures. And I know that one of the things that sometimes comes up for patients is they want to get this treatment, but they don't. It's almost like they have too little disease, right? And it's really hard to tell someone, oh, you do have this growing, but the spot's too small for us to inject it. So, there is that technical component that sometimes comes up, but we're pretty good now at knowing what we can and can't do. And so that's why we want to see everybody's films and not just the reports, so that we can review it with the team. So when someone's ready, what happens is they come back and they get a very, very light, what we call conditioning chemotherapy. And what that does is your immune system is really good at recognizing these cells when we give them back and saying, hey, these aren't our cells. These were messed with. They're different. And so we give just a light dose of chemotherapy to suppress your body from rejecting them. So to kind of allow your body to have them come in and do what they need to do. And that conditioning chemo is usually anywhere from 2 to 7 days before you get the cells back. That can be done outpatient, but you need to kind of stay locally when you're having that done. Then you come into the hospital, the cells are injected either into that pleurax, that pleural effusion, or into a tumor spot that can handle the volume. And we keep you in the hospital anywhere from, 2 to 5 days, depending on how you're doing, just for monitoring of any symptoms and side effects that emerge. And then you go home and you're basically followed very, very frequently, once a week in person and pretty much by phone every day or every other day for the first 30 days. And then it de-intensifies a little bit for the second 30 days.
So is this treatment given once or is it given multiple times?
Typically it's given once. There have been people on some of the other study that we did who had what we thought were good responses to the treatment, who we did go back and treat again, but it's something that the protocol sort of allows for, but it isn't a pre-specified part of it for everyone.
So Dr. Zauderer, in the beginning you said that you take some of the immune cells, and now we're at the time of COVID. Does this make us more vulnerable to infections and diseases that are out there and perhaps the COVID virus?
Yeah, I think that's a really good question. Not that we're aware of, because relative to all of the immune cells in your body, we're still taking out such a small relative percentage that it shouldn't, and we haven't had any reports in these studies to date of it impacting the immunity. Also, T cell CARs are very commonly used now in the liquid malignancies, both in trials and as standard treatments. And so if we take a step back and we look at all of those people that have the cells extracted, we don't see and we haven't seen any differentiation in how they're doing with respect to COVID and other infections based on participating in a T cell treatment. That said, when you get that conditioning chemotherapy, then your immunity is a little bit suppressed. And so that is a more tenuous time where you have to be very vigilant about your exposure to COVID and your exposure to anything else.
So patients then, basically what you're looking for patients who are in relatively good health. Does the disease have to be confined only to the pleural space, or can it be metastatic disease?
It can be metastatic disease. We have some data, in this space and from other studies that if we can incite an immune reaction where we inject it, that response can also be effective in other places. I think that the real sort of crux of what we're looking for, in addition to someone where it's technically feasible to give it, is someone where they can, sort of be okay with some of the hurry up and wait, I think, that often gets worked into participating in any of these trials. And so from the time at which the cells are taken, just the laboratory time, assuming there's no scheduling issues or hiccups, it's a 6-week minimum time in the lab. And during that time, you can get other treatments like chemotherapy, immunotherapy, radiation, if you need them. But what we don't want is someone who has rapidly progressive disease, because I think that's, there's just too much sort of waiting, I think, built into the pipeline that way.
Mm-hmm. And what kind of side effects do we expect with something like this?
That's a great question. One of the side effects that we're always worried about, but we haven't yet seen in the mesothelioma patients we've treated, is something called cytokine release syndrome. And that's when you give these cells back. They work so well, they stimulate the immune system so vigorously, it's overstimulated. And as a function of that, people can have issues with breathing, they can get confused. In the initial days when this technology was new, people got confused and even had seizures. And so we worry about an overwhelming immune activation. We have not yet seen that in the mesothelin studies that we've been doing, but that is a potential risk factor that we're always concerned about. The common things that we've seen are what you might sort of more typically expect, which is some discomfort where the needle is inserted, or if it goes into the fluid or the pleurax, some pressure from the volume that's being applied. There have been some low-grade fevers that people have had. And then in what happened in a bunch of people was about a week to 10 days after the treatment, some people had a fever. And we always looked at that as a marker that, the immune system was being activated. But that's also around the time when your blood counts are low from that conditioning chemo you got. So then you wind up with low blood counts, the T cells given back to you, and a fever. So it at least buys you a trip back to the hospital to have some cultures and make sure that you don't have any active bacterial infection. But those have been the things that we've seen.
So, Dr. Zauderer, so I guess you're looking for patients who, as I said, who are relatively healthy, disease that, that you can get to, either get into the pleural space or inject into a lesion. There seem to be, multiple options now taking place across the states. How do patients line these treatments up? I don't want to put you on the spot, but, I'm thinking, I get some questions. Should I go for immunotherapy or should I consider going on for a trial? Is there any information, or even if it's anecdotal, about how you would time immunotherapy around these T-cell trials? Is the thought process it's better before or better afterward, or totally unknown?
Yeah, I think that's a great question, and one that we all struggle with in figuring out how to counsel patients. None of us know. We don't know the optimal order. We don't know if there's some combined activity or synergy. We don't know if it's better to prime the system and then give the cells. We honestly don't know. There's a lot of people who have a lot of very strong opinions about these things. The challenge right now, I think, is with the Checkmate study and the approval of ipilimumab and nivolumab in the United States. Or not the problem, but the question has become much more complicated. And I think when I sit down and I talk with patients, we really work through what we think the likelihood of responding to something is. And the thing about checkpoint inhibitors, immunotherapy, is that the patients with epithelioid mesothelioma didn't get as much benefit from that. And so those are the people who tend to have the highest expression of mesothelin. And so it's sort of, at least for these types of T-cell studies, the people we think have the most of the thing we're targeting are also the people less likely to respond to immunotherapy. So it sometimes can make that decision a little bit easier. My basic philosophy about this study, any other study with me or elsewhere, is at the end of the day, and I don't mean this tongue in cheek, but there's no wrong choice. Whatever that feels right and feels comfortable to an individual. And if, you want to, after chemo, let's say you want to do a study, and then you can always get immunotherapy afterwards, that's fine. And if you want to get immunotherapy, and then if that doesn't work or stops working, then you want to look for a trial, that's okay. It has to sort of feel comfortable to you, because as much as there's uncertainty with any treatment, there's a ton of uncertainty with any clinical trial. And so it wouldn't be fair or ethical for anyone to say, I think this trial is better than doing a standard treatment. What I can say is, if you do a study, you can always go back to a standard treatment at the discretion of your doctor. But if you do a standard treatment, that trial may no longer be active, or you might no longer, be eligible for it. But, I never want someone to do something because they're afraid they're going to burn this bridge or that bridge. It has to feel like the right choice in the right moment for the person
Right.
doing
And I think
it.
with all trials, until we see a trial is completed and we have the data, it's really hard to say that one is better than the other. It's just that,
Absolutely.
I try to explain it as opportunities, to look at opportunities, but to also smartly try to line these treatments up so that this way you try to not knock out any, but you try to get a good lineup. So if one doesn't work, you're prepared to go on to the next.
Absolutely. And I think that,
So,
that there are also, other T cell trials going on that are directed to mesothelin. And so if you might not be eligible for one, but you might be eligible for one somewhere else. So there's a lot of things to consider on the national landscape.
Now, what about patients with sarcomatoid disease? Are there any T cell trials that you're aware of or that might be in development for a patient with sarcomatoid, or is it a problem finding a target?
It's more challenging with the targets, but there are actually some sarcomatoids, or and certainly biphasics, that have enough mesothelin to participate. And there's some preliminary data from Dr. Prasad Adesumilli suggesting that you may only need a small amount. And really what you're doing is you're honing the immune system to a place, and it doesn't matter if that mesothelin is 10% or 40%, as long as you have enough to sort of start the avalanche, that the avalanche will come. And we actually, in our initial trial, had a patient with biphasic disease who did very well.
So in terms of, determining responses, when do you expect to see a response after this treatment?
We're not entirely sure. On the trial that we have already completed, among the people who went on to respond, that response didn't occur for 4 to 6 months after the infusion of cells. And I think that actually speaks a little bit to how I think about sequencing the treatments, right? So you could imagine someone might get the cells, we might not see a response, and then the question is, well, what do we do? Do we give more treatment? If we give more treatment, what should that treatment be? And what I can say is that we don't think that immunotherapy after T cells impairs or interferes with the T cell activity. But chemo after the T cells will most likely wipe out the cells we just gave. So I definitely think if we need a treatment after cell therapy, I tend to favor an immune-based treatment, just because if something's going to happen with the T cells, it shouldn't necessarily interfere with that. And it may actually augment it.
And I guess that makes sense too, in terms of when you're choosing your patients, that if they don't have bulky disease and they're not galloping along, you have that time to wait it out. But if you have somebody with, sort of a life-threatening disease close to an organ, invading into an organ, then you're going to be a little more judicious about whether or not this would be the correct approach.
Absolutely.
So in terms of, I guess looking at these T-cell trials, and as you said, there are multiple trials taking place across the country. Are there great differences between these trials? Is it basically, are they all very similar? I just wonder, how do we look at them in terms of deciding this T-cell versus that T-cell trial?
Yeah. As is often the case, none of these things are compared head to head. And everyone's sequence that they insert is a little bit different. And so what we don't know is, well, does one sequence of alteration make the cells better than another? And we don't know that answer, and we're probably never going to know that answer. So the way I think about the cell trial options are in terms of the eligibility criteria. And to me, those are some of the differences. Is this given into the pleura? Is it given into the abdomen? Or is it something being given intravenously? And depending on the distribution of the disease, that may make more or less sense for a particular individual. And then, I think we also have to consider very, importantly, the logistics, when we think about the different locations and being displaced, and some places are going to be easier than others. Beyond that, it's hard to say that what the differences really are going to shake out to be between the different T-cell constructs that are being tested.
So, a lot of patients, ask about, how do they stimulate their own immune systems? Is there anything that they should be doing to get ready for a trial like this, or is there anything they can be doing to enhance what's happening in the trial?
Yeah, I get that question all the time too. And unfortunately, there's nothing that we know of that directly, that you can do that's going to enhance or prepare you. I do think it's important to, eat a balanced, healthy diet, to try to be as active in terms of, cardiovascular exercise as possible. We know that those things chronically contribute to inflammation in the body, sort of a bad diet and a lack of activity. So I think that the more that you do those things, we're starting to see some of the associations with long-term cancer outcomes. But beyond that, there isn't a magic sort of pill, formula, or bullet that we know of.
So in other words, they're really, dietary changes. We should all be eating a healthy diet, but any major dietary changes at this point, we don't think have any impact on mesothelioma or these type of trials.
Correct.
So, Dr. Zauderer, I think I recall at one point you were also giving Keytruda with this trial. Has that changed?
Yes, in the trial, the TARA trial that we're doing now, one of the things that we insert into the cells now is we wipe out their ability to express PD-1. And so the way that immunotherapy like pembrolizumab, nivolumab, ipilimumab work is what they do is that somehow the cancer is able to mask itself and suppress the immune system from responding to it. And rather than thinking of the immunotherapy drugs as stimulating the immune system, what you have to think about them is that they stop the inhibition. So you're not stepping on the gas, but you're taking the foot off the brake. And what we're doing in these Zauderer cells is there's no longer a brake. So they shouldn't be subject to suppression in the same way. So because of that, there isn't a plan to give any pembrolizumab afterwards because theoretically it shouldn't be needed to keep those cells around.
Which is great, which means that, you have a finite treatment and patients, then can go back to home, and it's not this, constant back and forth every 3 weeks for additional treatment at that point. So
absolutely,
they get to resume their life.
Absolutely.
And how long does it take? How long does it take the cells to recover? We talked about, you've taken out, some of the immune cells, and now you've reprogrammed them and you've given them back. following, the completion of this therapy, do they have an intact immune system? Or are they, pretty much, able to, fight off normal viruses the way the rest of us do?
Yeah, about 2 to 3 weeks after the conditioning chemo that you get in the days before the treatment, you should have a normal complement of your immune system back online.
Well, that's great. So I think this is, again, another, exciting opportunity. And how many patients will you be able to have, accrue to this trial?
That's going to depend on how people do and whether there are any serious side effects. But we anticipate somewhere between, 15 and 24 patients to be treated on this study.
And are you doing this in cohorts where you would do a few patients and stop and take a look again, so that patients may come to you and kind of get on a waiting list?
Absolutely. So we're doing it it's a phase 1 dose escalation. And so some of you may know already that's usually like 3 patients at a dose. And depending on how they do, it's either then 3 patients at the next dose or 3 patients at the same dose usually. And we sort of go from there. What we're doing is, screening people, collecting cells, freezing what we've collected, manufacturing, freezing if we need to. So, I think it's important when you meet anyone about any treatment, whether it's a cell therapy or anything else, to have a really good sense of what the potential timeline looks like. And then to work with your team to sort of figure out backwards if that makes sense for you. I think the second thing that I always talk about today, which a year ago I never would have mentioned, is, because you have to be admitted to the hospital, were there to be another surge and shutdown, some of these cases might get more spread out. We would by no means stop cell treatments, but we just might have less space and less beds available. We're all hopeful that won't happen in New York City again, but it certainly did last spring. And so that is another kind of variable in the mix. And frankly, that would be a risk, I think, anywhere in the country right now.
So, if we don't get that, that surge and things sort of remain somewhat what they are now, does the caregiver come to appointments? Can the caregiver be at the hospital with the patient, or, how opened up is it now?
So right now, a caregiver can come to appointments. We have visiting hours in the hospital for 8 hours a day, so 4 in the morning, 4 in the afternoon and evening. The one place that a caregiver can't come is to the actual infusion suite. So that day when the person comes to get that preparatory chemo, the person can come with them into the building, but into the actual infusion space, because people are too close together, right now, at least, you're not allowed to have someone back there with you, but they could be out in the waiting room or down in the lobby or something like that. Obviously, if someone's sick or not feeling well or needs help, or there's a language barrier or a physical mobility barrier, we make exceptions to those policies.
Okay, and so patients now who'll be coming from out of town, they'll need to find lodging. Does Memorial Sloan Kettering, do you have relationships with hotels or places where patients can stay?
Yeah, so in New York City, there's a place called Hope Lodge, which is run by the American Cancer Society, and a patient and a caregiver can stay there. And that's something we work with people to coordinate. We also have relationships with several hotels. It's still New York pricing, but it's a relative discount from that. And then some people have actually worked out short-term sublets in the area near the hospital, and those are things that we have some connections and network to help people navigate. Mostly our social workers help with those components.
Right. And, from the foundation standpoint, we will help, when there's a financial need. We have the travel grant program, which will help with lodging and with transportation when necessary, because we're committed to making sure that, patients can get to a trial and then also stay in the trial, that they, financially they have some cushioning. So.
Yeah, that's been a
Dr.
great
Zauderer,
resource for patients over the years.
So, I think this was a good discussion about these trials. Is there anything else that you wanted to discuss while we're on the call, or anything that I didn't ask you?
No, I mean, I guess the only thing I would say is if, people watch this and hear about it and they're interested, these days we can do a lot of the initial screening through virtual visits and sending records and see if it's even a potential possibility to even make it worth coming to see us in New York. So we're always happy to do a remote, start with a remote review and take it from there.
Okay. Well, thank you very much, Dr. Zauderer, and I really appreciate your time today. Take care. Stay safe. Thank you.
Thank you, Mary. You too.